So good evening and welcome to Basic Science Lights the Way. Tonight we'll tackle some of humanity's biggest open questions like what are we made of? Are we alone? And what is the nature of the universe? Our researchers will weigh in on how each of them are chipping away at these mind-boggling mysteries. UC Berkeley excels at asking and investigating these fundamental questions. Our faculty experts demonstrate how broad existential ideas that have confounded humanity for millennia can be approached through precise experiments. What you hear tonight may sound like science fiction. So did genealogy, black holes, radio waves, and nuclear fission before their time. Only by asking the greatest questions can we make the most significant discoveries. So please get ready as we explore the thought-provoking frontiers of science. And now it is my pleasure to introduce our moderator this evening, Dipti Nayak. Dipti Nayak is an assistant professor in the Division of Genetics, Genomics, Evolution, and Development in the Department of Molecular and Cell Biology. Her research group uses genome editing to study archaea, which are single-celled microorganisms and also some of the oldest life forms on our planet. Her group specifically focuses on archaea that produce and consume methane, which is one of the greenhouse gases causing our planet to warm. Thank you for being here this evening as our moderator, Dipti, and let me turn it over to you.
Thank you for the introduction, Dean Kahn. And first and foremost, I want to congratulate you being elected to the National Academy of Sciences today. That's really, really great news. And now I'm happy to be here with all of you this evening. So as Dean Kahn mentioned, one of the big open questions in biology that we're all trying to study is how life originated. As Dean Kahn mentioned, I study organisms that belong to a domain of life called archaea. Some of these archaea produce methane gas and are called methanogens. And methanogens have consistently been some of the most important members of the biosphere for the entire history of life on Earth. From their central role in the emergence of life billions of years ago to their contributions to some of the most pressing issues that are facing modern human society. In the context of the topic today, what's particularly interesting about these microorganisms, these methanogens, is that they are the progenitors for humans and all the life we see around us. As we learn more about these organisms, we learn more about ourselves, we learn more about our planet, and we also learn more about how life on Earth has evolved over billions of years. And also, in the context of what we're going to discuss today, if life is found in the liquid water oceans of the icy moons of Jupiter or Saturn, it's likely that organisms quite like these methanogens that my lab studies might be there too because of the similarity of the geological processes that are found in these places. And further, from the perspective of our own planet, if you understand more about how these methanogens, these archaea, make methane, maybe we can find solutions for our anthropogenic climate change that is being caused by their activity in agriculture and waste production. So that's how my work fits into the context of the big questions that we're going to talk about today. And now I'm going to move to our first speaker this evening, Professor Craig Miller. Craig Miller is the Judy Chandler Webb Endowed Chair in the Biological Sciences. And he's also the professor and division head of Genetics, Genomics, Evolution, and Development in the Department of Molecular and Cell Biology. His lab studies how patterns form during development and how they change during evolution. And his lab specifically focuses on the vertebrate head skeleton and uses genetic approaches in the three-spine stickleback fish, a species complex that has repeatedly evolved head skeletal adaptations. Broadly, they seek to understand the genetic basis of evolved differences in morphology. Greg, thank you so much for joining us this evening, and could you share some of your research with us?
Sure, be happy to. Thank you, Dipti, and congratulations, Dean Kahn. So as Dean Kahn mentioned, one big open question in biology is what are we made of? And since are programmed to make humans is encoded in our genome, this question could also be rephrased as what does non-coding or what's been called junk DNA do? And I want to tell you about that. So the central dogma of molecular biology describes how information that's encoded in the DNA in the genome is transcribed into RNA, which is then translated into proteins, which then accomplish the things that the cell needs to do. While all cells and organisms in general have all the same DNA, only a small subset of that DNA is transcribed into RNA. And many genes are transcribed differently in different cell types. And so one fundamental biological mystery is how cells know what subset of the DNA they need to transcribe into RNA. One surprise came upon the sequencing of the human genome that revealed that our 3 billion base pair human genome only contains 1.5% of protein coding genes, which is this tiny little red sliver here, which means that 98.5% of our genome is what was called at the time junk DNA, which was presumably not having function. However, what this junk DNA remains a really outstanding mystery and is quite controversial. Some biochemical data suggests that 80% of our human DNA genome participates in at least one biochemical event, but yet only 5% of the human genome is evolutionarily conserved. And so one fundamental mystery is trying to figure out what this non-coding or junk DNA does. And what's clear and what I'll tell you today is that this junk or non-coding DNA regulates the transcription of genes in embryos or organisms in space and time. And this regulation is absolutely critical, both for disease, as this non-coding DNA is the primary source of disease in humans and also trait variation in humans. And it's also critical as it's the primary source of evolutionary change in natural populations. And so the transcription of genes, as I mentioned, is regulated in space and time. And so within organisms or multicellular organisms, you often see restricted patterns of transcription. And so work in many, many organisms, including fruit flies, fish, mice, and humans, has shown that the decision of cells to transcribe genes is precisely encoded in the DNA. Shown here is a Drosophila melanogaster, or a fruit fly embryo that is a very young embryo that is sort of formless and shaped as a football. In blue is labeling the DNA in the nuclei of every single cell. And so all cells have DNA, whereas in magenta is stained for the expression of a single gene called even skipped that appears as these magenta stripes. And so this even skipped gene is regulated in space and time, in that in space, it's turned on, or the DNA is transcribed into RNA only in these cells, but not in the cells that lack the transcript. And this is a major mystery of why this gene is transcribed in some cells, but not others. And work for decades has shown that the information that is encoded in the fly genome maps to non-coding regions around the exons of this gene. And furthermore, each stripes are regulated independently. For example, there's a very well-studied non-coding DNA that is responsible for driving transcription of this second stripe of expression and not expression in any of the other cells. And so this non-coding or junk DNA is understood biochemically to function in regulating gene expression by containing what are called enhancers. So enhancers are regions of DNA that positively regulate gene expression or transcription, often in a spatial and or temporal specific manner. These are also called cis-acting regulatory elements. And enhancers, like in this cartoon, are known to bind transcription factors, which are colored in green in this cartoon. And then the intervening DNA loops out while transcription factors binding the enhancer physically activate RNA polymerase bound at the promoter, resulting in the production of messenger RNA from the start of genes of interest. These enhancers can be tested experimentally for function in organisms by testing their ability to drive transcription. And these experiments usually use what are called reporter genes, which are genes that are simply easy to visualize. For example, green fluorescent protein, which you can see when it's translated as green fluorescence. And so candidate non-coding or junk DNA elements can be ligated upstream of a minimal promoter and a reporter gene. And then those constructs can be injected into one-cell embryos to make transgenic animals that incorporate this DNA sequence into every cell in the organism. And then if these candidate enhancers have enhancer activity, part of the embryo or animal will light up. For example, cartooned here is a single stripe in the fly embryo, like of the 7 stripes I just showed you. So this can functionally define enhancers and shows that non-coding regions are sufficient to drive transcription of genes in space, but also in time. As embryos develop in time, you see that enhancers often turn on or turn off. My lab studies the evolutionary morphology using this fish model system shown here, which are 3-spine sticklebacks. Evidence from this system overwhelmingly supports this idea that junk DNA is not junk, but is important for critically regulating the evolution in nature. And so this species complex has underwent an adaptive radiation where ancestral forms that live in the ocean are cartooned in the center. And then like the Galapagos finches, they have repeatedly adapted to new freshwater environments, in this case, lakes and creeks drawn on the outside, including Strawberry Creek on campus here, which you should recognize the storm drain picture from the Berkeley campus. Okay, and so these forms are genetically crossable, and so we can cross the ancestral oceanic forms and derive freshwater forms to directly map the genetic reasons of why certain morphological traits change. Dramatic changes evolve in this system, which are shown here. And so these are micro CTs or 3-dimensional X-rays of adult fish caught from the ocean on the top and from a lake on the bottom. So the ancestral form is covered in bone, while the freshwater form has lost a lot of bone. Extensive genetic studies in this system have identified specific genes and genetic changes that underlie evolved traits. There's been 3 loss traits that have been studied exhaustively that include the loss of the fish's hind limb, the loss of the armor plates on the fish's flank, and reductions in pigmentation in the skin. All 3 of these traits have been mapped to specific genes, and in all 3 cases, the underlying mutations that affect the genes are thought to be regulatory or changes in the non-coding DNA that affect where and when in space and time these genes are transcribed. And these genes do not have mutations in their coding sequence. And so here, these major adaptive traits are regulated by changes to this non-coding junk DNA. Those are all loss traits, but are there any morphologies that are gained in freshwater? There are. My lab here in the past 15 years has studied 2 gain traits shown here, where ancestral forms shown on the top evolve more bone. And so on the left is showing you ancestral field of teeth that evolved to be much more numerous in freshwater, which is thought to be adaptive for a shift in diet of small prey in the ocean to larger prey in freshwater. Similarly, bones in the skull quantitatively get longer in multiple freshwater populations. And my lab has shown here that both of these gain traits evolve not via coding changes in exons of genes, but instead due to changes in the regulation of these genes that affect where in space and time they're transcribed in fish embryos and post-embryonic fish adults. And interestingly, in both cases, our data shows that these gain traits are due to reductions of expression of what we think are repressors. So we think that the fish can evolve more of something by reducing repressors. Okay, so here these gain traits are also due to changes in this junk or non-coding DNA. Shown here is a summary of all the genomic changes that accompany freshwater adaptation in this system. There's over 100. This has been exhaustively studied by whole genome sequencing in multiple populations. And the summary shows that a minority of those changes are in coding sequences or the exons of genes, while the vast majority map to regulatory regions that are thought to affect the regulation of transcription of genes in space and time. This exact same conclusion holds for human disease. And so in humans, most disease-causing mutations are in this junk or non-coding DNA. And it's summarized here. And so shown, for example, for 2 types of cancer, prostate cancer, breast cancer, but also lupus and Crohn's disease. The vast majority of mutations associated with those disease phenotypes in humans are cartooned purple here, which are regulatory and are not mutations in the exons of coding genes, but instead are due to what are thought to be regulatory variants. Shown here is an overall summary of not for specific diseases, but all human disease and trait variation. And the conclusion is the same, that the vast majority of genomic variants that are associated with disease and variation are regulatory and mapped to this junk DNA. Okay, so most human disease and also non-disease variation in humans is due to mutations in the regulatory regions. And so I think that this is telling us that junk DNA is functional and that junk or non-coding DNA regulates the transcription of genes in embryos or organisms in space and time, and that this non-coding DNA turns out to be absolutely critical, and it's the primary source of both human disease and trait variation, but also the primary source of evolutionary change in nature. And so from this, I hope you're convinced that it's not junk. It instead is super important for disease and evolution. 3 outstanding questions about junk DNA include, where do these enhancer sequences come from? Why are some enhancers conserved and others not? And how do enhancers regulate transcription in space and time? And lastly, I'd like to thank the people in my lab who did the work, including current graduate students, undergraduates, and postdoctoral scholars, and past students. And I'd also like to thank the National Institutes of Health and National Science Foundation for funding. And thanks. Be happy to stop and take questions.
Thank you, Craig. It looks like we have a few questions for you. So let me begin with one till we get questions from the chat. The first question that I have for you is, so as you mentioned right at the end of your talk, where do enhancers come from? So what is kind of the current theory around that?
Yeah, that's a great question. Still an active field of research. There's some really good evidence that some enhancers are born from transposable elements or jumping genes. And that's interesting because those are mobile elements that can jump around the genome. But it's a small fraction of vertebrate enhancers, at least, that are thought to be derived from transposable elements. Most enhancers are not and are thought to evolve de novo from genomic DNA sequence. And there's not been much work or understanding on that, but there's some ideas and a little bit of data that maybe enhancers first evolved as what are called proto-enhancers, and that maybe if the DNA sequence can first acquire mutations that biochemically open up the chromatin, that could then allow subsequent mutations that if they happen to make a mutation that gains a transcription factor binding site, that then the loose chromatin can then gain the ability to turn on transcription. But I think that's a really exciting question, and I think we now have so many sequenced plant and animal genomes throughout the tree of life that we can really precisely date when most enhancers were born and where on the tree of life they arose. And I think future experiments will determine where they came from.
That leads me to the next question that came from the chat by Robert Branson. And Robert asks, what is the relationship, if any, between the junk DNA and epigenetic effects? Do you have any thoughts or any examples along these lines?
Yeah, that's a really great question. And I think in general, epigenetic effects affect the regulation of genes. And so I think they're very intimately related. And epigenetics refers to modification of the DNA that usually involves histone modifications, where the histone proteins that the DNA is wrapped up can be methylated or acetylated, and that can either condense or open up chromatin. And yeah, I think they're very closely related. And I think the epigenetic effects that have biological effects are usually because they're modifying enhancers, either opening or closing them.
Right. And so the next question is more about what we should consider as being junk DNA. So this question, starts with the idea that, DNA that's involved in regulation, such as enhancers, should we even consider that as junk DNA? And if that's the definition we're working with, what fraction of DNA would not be considered either regulatory or coding? And how would we even where do we draw the line between junk and functional in some sense, right?
Yeah, that's a great question as well. And I think there's a real sort of mystery in the field that I mentioned. The biochemical data shows that 80% of the human genome participates in some biochemical event, but yet only 5% of the sequence is evolutionarily conserved. But what's confusing,
Right.
some enhancers are conserved, but others are not. And so just because the DNA sequence is not conserved certainly doesn't mean it's not functional. So for example, there's very little non-coding conservation between fish and mammalian genomes, whereas the exons are super highly conserved. But yet there's lots of cases where mammalian enhancers can function in fish and vice versa, despite the absence of sequence homology. So I think conservation is not a good metric for function necessarily. And I think the biochemical data that 80% was spread across over 100 different cell types and lots of experiments where cells were treated in culture. And I think the DNA itself is pervasively transcribed at low levels. And I think most people think that that's noise, but it's quite difficult to experimentally address that. So if the genome is transcribed at a very low level, does that have any function? I don't think so. So my gut feeling is the true functional non-junk DNA number would be somewhere in between 5 and 80. But I don't really have any better guess which side it would be towards. I think it's still early days.
Okay, and then expanding on that idea of the connection between, junk DNA and DNA that, and disease. Do we know, for example, where mutations in junk DNA may lead to hereditary diseases? What's the state of the field? And diseases that are just hereditary diseases. So anything you inherit, a disease.
Yeah, the bulk majority of human disease are due to mutations in regulatory regions. And I tried to summarize that at the end, but it's something like 80% of disease-causing mutations are thought to be in non-coding regions. And I think about half of those are intronic and a half of those are in between genes. But we know a lot of enhancers aren't introns, and the intronic mutations might affect splicing or other aspects. But for sure, there's thousands of diseases that have been linked to specific enhancer variants. Basically, every disease, every common disease that you could think of. Cancer for sure. Most diseases that confer risk to cancer are mapped to non-coding regions. But I think across the board, every single common human disease has examples where regulatory mutations contribute to the disease risk.
Okay. Thank you so much, Craig. I think in the interest of time, I'd like to move along to our next speaker, and we can come back with more questions at the end. And so moving along, we will turn to our next speaker, who's the chief scientist of the Berkeley Search for Extraterrestrial Intelligence Research Center, also known as SETI. And this is Dan Werthimer. So Dan Werthimer is the co-founder and principal investigator for the enormously popular SETI at Home project, which more than 8 million members of the public have used to process data collected by radio telescopes searching for emissions from other intelligence beyond our planet. He directs other UC Berkeley SETI searches at radio, infrared, and visible wavelengths. He is also the principal investigator for the worldwide collaboration for astronomy signal processing and electronics research. He has taught courses at universities across the globe, including China, Hungary, Zimbabwe, Uganda, and Kenya. He has published a large number of papers in the fields of SETI, radio astronomy, instrumentation and science education. And he is also the co-author of SETI 2020 and editor of Astronomical and Biogeochemical Origins and the Search for Life in the Universe. In addition, he's received the precious Frank Drake Prize of SETI Institute in 2021 for his work. Dan, we're so excited to have you here, and please tell us about your work.
Thanks very much, Dipti. I want to talk about this question, are we alone? Is anybody out there? And so one of the questions when you ask this, anybody out there, are there planets out there, and are there any good planets where there's life could be? This was pioneered at Berkeley. It turns out it's really hard to find planets because they're little dinky things. 1,000,000 Earths could fit inside the sun. They're right next to these really bright stars. So they're found indirectly. You can't see the planets, although we're starting to be able to do that. But the indirect methods that were pioneered at Berkeley are that when the planet goes around the star, it pulls on the star and the star wiggles a little bit. So if you see a wiggling star, it doesn't wiggle much, but that betrays the presence of a planet going around that star. Now, thanks to the work at Berkeley and others, we found that there are about 1 trillion planets in the Milky Way. And if that's not enough for you, there's 100 billion galaxies like the Milky Way, a lot of planets. And we're beginning to learn about the number of good planets. The technical term is in the Goldilocks zone. So these things in the green zone, not too hot, not too cold, is too hot, too cold. So there are a lot of good planets. Another question you want to ask about Are We Alone is, does life get started? If you have a good planet, how often does life get started? And people have done experiments where they simulate the early conditions on Earth. And what you can start with these very simple chemicals, and you can get amino acids, and recently people got nucleotides, the things that make up DNA. So we're beginning to learn how the life gets started, and we're optimistic about primitive life. It might be all over the universe because it happened very quickly on Earth. The oldest rocks you can find, just as soon as the Earth cooled down, life popped up. So because it's a fairly quick process, we think it's going to be, life is probably going to be abundant, at least primitive life. And there may even be life in our own backyard. This moon going around Jupiter called Europa has a warm ocean, this blue stuff. It's unfortunately covered with ice, so it's hard to see if there's something swimming around down here. But there's another moon, Enceladus, going around Saturn, and it's got a warm ocean, but there are cracks in the ice. So the water is squirting out its geyser, and NASA and the Europeans are thinking about flying a spacecraft through the plume and sampling that water to see if there's some primitive life in that water. So SETI is the search for extraterrestrial intelligence. That's what I work on, my research, my students work on. I get the credit for their work. So there have been a lot of ideas for how to communicate with ET. Gauss 200 years ago said make a large structure, triangles and squares. ET would see that we knew about the Pythagorean theorem. Von Littron said dig a large ditch and fill it with kerosene, make a bright circle of light. ET would see this and get in touch. Charles Crow said use mirrors to reflect the sunlight to the Martians, several mirrors, in the shape of the Big Dipper, and ET would see these bright lights and get in touch. More recently, 50 years ago, NASA put a plaque on the Pioneer 10 spacecraft that's got a couple of humans and a picture of the solar system with the sun and Mercury, Venus, Earth, and here's the spacecraft leaving the Earth. So one of the ideas in SETI is that Earthlings are sending off a lot of television, radio, radar signals, navigational beacons are leaving the Earth, traveling the speed of light. This is television power leaving the Earth. And the Earth is brighter than the sun now at television frequencies. The early shows like Ed Sullivan, I Love Lucy, has gone past 10,000 stars. The nearby stars have seen The Simpsons. So maybe ET's doing that. We've even sent intentional signals. This is controversial, but the idea is maybe they don't speak English, but maybe pictures would be a good idea. There's a picture of a human, DNA, a solar system. So pictures might be good. I don't think this is a good idea. Maybe ET will come and eat us, but we are sending off signals all the time that are unintentional. My main motivation for looking for ET is that in order to park in front of the astronomy building, you need a Nobel Prize. It's really hard to park on campus. So if I find ET, I might get this prize. So we want to use these big radio telescopes to look for radio signals from ET, maybe TV or maybe some GPS signal or something, radar signal we could detect. And this was the first one we used in Northern California, Berkeley telescope. And while we were using it, this is what happened to it. This is the telescope down here. So we moved to this telescope in West Virginia. And while we were using this telescope to look for ET, this is what happened to that telescope. Then we moved to this telescope. This is holds 10 billion bowls of cornflakes. And while we're using this telescope, this is what happened to that while we were looking for ET. And you might ask, why does this happen to Dan? The World Weekly News said that aliens did not want to be discovered, and so they zapped these telescopes. So one of the problems in looking for these radio signals is it takes a lot of computing power. We don't know what channel, what frequency to look at. We look at millions of channels. Here's a strong signal at one of those channels. We also want to look for drifting signals that, or we want to look for pulsing signals, bip, 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 and that takes a lot of computing power. So as Dipti mentioned, we launched this project called SETI at Home, where we asked volunteers around the world if they have a laptop or a desktop or a cell phone to help us analyze data. We have 20 years of data, a few petabytes, which we stored at Berkeley's NERSC computer. We broke it up into little pieces, and everybody got a little piece of the sky to analyze with their home computer. They ran a SETI at Home screensaver, and if they're the lucky ones that finds ET, they get the Nobel Prize in the parking spot with me. There were 8 million people, though. You might have to share it with 8 million, probably just I think the Nobel Prize is only 3 people. They were in about 200 countries. For many, many years, it was the world's largest supercomputer. It made the most sensitive search that Earthlings have ever done. What we're doing now is not asking the volunteers for so much help, but we are going back to the most interesting things that the volunteers found. We have the 200 best candidates. We're using this big telescope because the Arecibo telescope fell down. This is a telescope in China, 500 meters across, and we're looking at these 200 best candidates. Well, we haven't found ET yet, but I want to tell you about some of the things we found along the way. We developed a curriculum with Lawrence Hall of Science in Berkeley to just because a lot of kids are interested in this question, are we alone? It touches on a lot of different fields in science, a great way to get kids interested in science. We developed some open source software so that you can do lots of different kinds of scientific computing at home. You can work on global warming, you can do gravitational waves, black hole projects, protein folding, COVID-19 cancer drugs, all kinds of therapeutics. There's about 100 projects. You can pick your favorite projects and use your spare computing cycles. We call it the democratization of scientific supercomputing. Also, another thing that happened was that the instruments that we built for SETI got used for a lot of different kinds of astronomy, led to all kinds of fun discoveries. The first images of black holes, weighing the universe. We just learned using SETI instruments that the whole universe is jiggling around like jello due to a background of gravitational waves. A spin-off that I'm sort of proud of is that we've had 100 students work in our group over the last 40 years, and they've gone off to do a lot of amazing things. One of the new things that we're working on is called the PanoSETI transient search. And we want to look for not radio signals, but visible signals, optical signals. And we want to look for flashes of light, like a lighthouse flashing, looking maybe from a laser signal that ET would have. And the problem is we don't know where that flash is going to go off, and we want to cover a big part of the sky. So we want to build a thing with lots of telescopes looking at different parts of the sky so we can maybe catch one of these flashes if they're out there. And we don't have a thing with lots of telescopes yet. We're trying to raise the money for that, but we have some telescopes. So we have 3 telescopes at Lick Observatory, which is operated by the University of California near San Jose. And we're building some more at Palomar. And I'm really excited about this project because we're looking at a whole new space that astronomers and scientists haven't looked for. We're making a movie of the sky at 1 billion frames a second, so we can look for very short flashes, 1/1000th of a second long or 1/1,000,000th of a second or now. Nobody's really explored the sky at these kind of timescales. And whenever you do something in astronomy where you're looking at a new size scale or a new wavelength, or in this case, a new timescale, it seems like nature has phenomena to be discovered. And so maybe we'll find something interesting with PanoSETI. So we haven't found anything yet, but I'm kind of optimistic in the long run if ET's out there that Earthlings will discover them. And the reason I'm optimistic is because even though we're just getting in the game now, we're just learning and we're an emerging civilization, the radio telescopes are growing exponentially, computing power is growing exponentially. We used to listen to 100 channels, now we're listening to 100 billion channels. And so I think if that exponential growth keeps going, we will be able to answer this question, are we alone? And a couple of my favorite quotes, Phil Morrison said that SETI is the archaeology of the future. We find ET, they're probably 1 billion years ahead of us. We're kind of a middle-aged star. A lot of stars are quite a bit older. So we could get on the galactic internet, learn what's in our future, how did they survive, get through their bottlenecks when they were killing each other. Carl Sagan said that SETI is profound either way. And what he meant by that is that if we find ET and get on the galactic internet, we could learn a lot, all their music, poetry, literature, science, medicine. But it's also profound the other way. If we can do a thorough search, maybe in your lifetimes, and we don't find anything, that's also very profound. It means that life is incredibly precious and rare. We better take really good care of our planet and the life on it. If you've been asleep, this is kind of a summary slide. No ET so far. We're still working on it. Thank you very much.
Thank you so much, Dan. That was fascinating. So we have a few minutes for questions. I'll start with one from the audience. Do we have any estimation of the percentage of planets that have orbital periods that are greater than 8 of our years? So this is from Dave Almond Smith.
So this is a difficult question for astronomers to answer right now, because the ones that take a long time to go around their star, it's hard for us to find them. So we think there are probably a lot of them, but there's probably it's kind of an observational bias problem. We could start to see the ones that are going around every year or two, but we don't have a lot of planets that are going around, like Jupiter and Saturn, that take many years to go around. The other thing that's kind of missing is we don't have a good inventory of the little dinky planets like Earth. So the big ones like Jupiter and Saturn are really easy to find. We know there's a zillion of those. We think there's a zillion Earth-like ones, but we're just beginning to explore that population. And Berkeley's kind of one of the leading teams on that.
Okay, so I have time for one quick question before I have to move on. And I think this is a really pressing one given the age that we live in. Do you have any thoughts on how AI might affect your search in the future?
Yeah, so the problem with SETI right now is that we kind of look for, it's very anthropocentric. We look for things that Earthlings are doing. But if you look at what happened 200 years ago, and people were saying they didn't have radio and they didn't have lasers, so they were talking about fire and mirrors and things so it might be that we're doing the wrong thing. But and we kind of look for signals that we're transmitting. We look for things that look like FM or TV, but what if it's not some kind of signal like that? Can we think outside the box? So these AI and machine learning tools, they do something interesting, or they can do something interesting called anomaly detection. So instead of looking for things that we tell the computer, look for this thing, a pulse thing, or look for that kind of thing. What if it's not one of those? They can just say, hey, I found something interesting. I've never seen something like that before, and alert us to it. So I'm excited about that kind of anomaly detection.
Okay, thank you again. And we'll come back for more questions right at the end. And so with that, I want to move on to our final speaker of the evening. Please welcome Yasunori Nomura, who's a professor in physics and also the director of the Berkeley Center for Theoretical Physics on campus. Yasunori Nomura works on theoretical physics, in particular particle physics, cosmology, quantum gravity, and quantum information. His current research focus includes the black hole information problem, holographic descriptions of spacetime, and also multiverse cosmology, which we're going to hear about today. One of the most notable things his work has done is that he's brought a new connection between quantum gravity and quantum information science through the concept of holography. With that, I'd like to welcome, with that, I'd like to introduce Yasunori, and thank you for joining us. Would you like to share your research with us today?
All right. Let me just start and thanks and congratulate Dean Kahn on being nominated in National Academy of Science. That's great. So what I'm talking about today is something called multiverse. You may have heard in the movies and so on. So the idea that our universe, entire universe, may be one of the many types of universes. And I want to start from the mentioning that this idea came from by looking into our own universe and try to understand certain features of our own universe. And in fact, this multiverse idea has to do with the unification of two pillars of modern physics. quantum mechanics on one side. This is the theory needed to discuss like very small objects like atoms and nuclei and so on. And general relativity, which is needed to discuss gravity and big space-time effect, the black hole, and then our whole universe. And these two theories are very, very successful in each domain. But it's very hard to unify, namely have a theory containing both inside that master theory, despite the fact that our world is quantum mechanical and have a gravity. So we definitely need that. And this is one of the biggest problems our theoretical physicists are facing right now. And the multiverse has to do with that question. So to discuss multiple universes, we really have to know what our own universe is first. And in fact, this cosmology of our own universe progressed a lot in past 100 years. And now it's in precision science. So universe is expanding. That fact was discovered about 100 years ago at the time of Einstein. And then this fact means that because the space is expanding, universe was hotter and much denser and shining early on. So that's why the name Big Bang you may have heard. And this one, we can see that now when the universe was very, very, very young, which is 380,000 years old. Currently it's 13.8 billion years old. How? Because the speed of light is finite. If you look at the distance, like the distance where the light takes 1,000 years to travel, then we're looking at the universe 1,000 years ago now, because it takes 1,000 years for light to come to you. So in that sense, if you look back farther and farther and farther, you're looking back more and more and more, and you can see the shining Big Bang universe. You must be able to. But the night sky is, of course, not shining for you. But yeah, in fact, it's shining if you look at in microwave, because of the wave from early on, it's just stretched. And in a microwave range, we have a map of the universe, a very, very early universe. And that universe is almost perfectly uniform and only small fluctuation of densities, 1 in 100,000. And we can even simulate the starting from those kind of small fluctuation, put that in a computer, and then you see that this denser region become more dense due to gravity, and the thinner region becomes thinner, and our structure forms. And in fact, we can go back further, and we can even calculate what happened when the universe was 1 second old. In those times, and the various nuclei like hydrogen, helium, lithium, and so on was synthesized, which we can calculate the relative fraction of the relative numbers of these elements, and that precisely agree with the current observation. So we really understand the universe back in 1 second old, okay? And even this small fluctuation, we can have an idea, which is called cosmic inflation, and universe expand like crazy, and in a fraction of a second, 1 second, the size of the nucleus will become the entire size of the currently observed universe. And that theory will explain this fluctuation origin of the galaxy as a quantum fluctuation back then, when the universe is 0.00001 years old, okay? And that prediction and data agrees this well. This is in a lower right corner. So it's a precision science, okay? So then what's the problem? Yeah, there is a problem. That's what discovered in 1998 by Saul Perlmutter, our colleague at Berkeley, and his colleagues, or in fact his competitors. And what they discovered was that expansion of the universe is accelerating now. Okay, it's becoming faster and faster. That's impossible, because gravity is a universal attractive force. So if there's only matter, whatever that matter is, the expansion must be decelerating. In fact, they were trying to measure how fast expansion was decelerating, and they found it's actually accelerating. Yeah? So that means that what they found is not energy density of matter. In fact, we knew that what can cause such kind of accelerated expansion of space, it's called vacuum energy or dark energy. This is the energy of space itself. It's not matter. Even if there was no space, vacuum or space can have energy, and that can cause accelerated expansion. So what they found was that. Okay, but the crazy thing is that if you want to estimate this energy of the space using the current theory, quantum mechanics, and general relativity, and that's 120 orders of magnitude bigger than what they found. That's why we thought that such a thing doesn't exist. Okay, so it's just not there. That's some artifact of the theory or something. But now we found it. Yeah, that's way smaller than what naive theory would tell you. And in fact, it's even more mysterious that the current energy density of the universe, 70% of this dark energy or energy of the space, and only 30% of the energy density of matter, average energy density of galaxy and stars and so on. So it's comparable. It's only 1:2 ratio. Okay? That's crazy. If the universe was keep expanding, if universe expands, the energy density of matter becomes small because energy density is energy divided by volume. If the universe volume becomes bigger, it's becoming smaller and smaller and smaller. But this energy of the space, even after expansion, looks space. So it's constant in time. Okay? So early on in the history of the universe, this energy density of space must have been tiny, completely negligible. But it must have a mechanism to keep this tiny energy density of space to be almost negligible, but not to the zero in such a way that after 13.8 billion years later, human make observation, then it's comparable. Completely crazy. Okay, so we just stuck by this observation. It was a huge impact, and they got the Nobel Prize in 2011, so they can park, or he can park in the parking spot. But if you look back at the theory paper, back then already in 87, again, Nobel Prize winner Steven Weinberg discussed the following. Okay, the people couldn't find a mechanism to set the energy of the space to be zero. Okay, that's why we just believe, oh, it's not there. Okay, we can't find a mechanism. So he said, oh, because we couldn't find it, let's just suppose that there is no such mechanism. Then what happens? Of course, there is no such mechanism, then the energy of the space is huge, okay? 100 order of magnitude, 120 order of magnitude bigger. But if you have a variety of a universe, even if the natural size is huge, if you have so many different types of universe, accidentally some of them may hit the small vacuum energy density. And what he showed in his calculation is unless that's the case, unless it's comparable to the current energy density of matter, nothing will form. No galaxy, no star, of course, no you, okay? Because a universe was a soup, okay? But before soup become galaxies, energy of the space dominates it, and then soup is diluted, period, okay? Nothing happens. So these lucky universes is the only place any observer can be there, okay? And then those observers will look around and make a measurement and will surprise why energy of the space is so small. This is what he said. And in 87, when the people believe that such energy density doesn't exist, he said that, okay, if this were a mechanism, it's very unlikely to be exact zero. So therefore, you will find this energy density of space, which happened 10 years later. But this is a wide assumption. And even you think, oh, is this only evidence? No. If you further look back, some theory called string theory, which is the only candidate we have to unify this quantum mechanics and gravity. And that theory predicted that the space is not three-dimensional. It's a higher dimensional. It's 9. So that means that the 6 of them is very small. So it's like this sheet of paper. Sheet of paper looks like 2-dimensional, but you have thickness. So that thickness is the 3rd dimension. Our world is like that. You have a thickness in 6 extra dimension. And then if these 6 extra dimensions have a different shape, it can take different shape, then it looks to us it's a different universes. So this theory already contained in 80s a mechanism of creating so many different universes, including different space energies and so on. And these universes keep creating, if you solve the equation, and in the form of the bubble in many universes, we are one of them. So this picture is what we call a multiverse, the summary of we keep learning that we are the tiniest substance. But we reach this even multiverse picture based on observation and theory. And this is a key portion of unifying quantum mechanics and general relativity to pillars of modern physics, which I'm doing together with my colleagues at Berkeley Center for Theoretical Physics And many of them are not working on multiverse, but it's various features of the universe. And then we appreciate your help if you could do any. Thanks.
Thank you so much, Yasunori. So we have a few minutes for questions. And we have a question from the chat by Nolson. And the question is space-time doomed?
Oh, good. Yeah, the space-time, in some sense, it exists, of course, right? But if that exists or not, fundamentally is a different question. Probably space and time is emergent. For example, does the concept of water exist? Continuous water. It's not. If you just divide it, it becomes H2O molecules, like this Mickey Mouse thing, okay? But we describe as a continuous water, okay? The space and time will be like that. So in that sense, you could say it's doomed, it doesn't exist. Whether it exists, you have to define the word exists, okay? So in that sense, it's not probably fundamental, but it certainly exists in the usual sense.
That's great. And so one question I have about your talk too is maybe connecting it to the previous talk is, we're thinking about searching for extraterrestrial life on other planets or beyond Earth. And so like, what about the concept of life that goes beyond the universe and into other multiverses? How do we even go about looking for something like that?
Yeah, that's great. But it looks like in our own universe, again, the similar things happening. Why our planet is so well designed? We know now it's not well designed. So many planets, most of the place, Jupiter, how come we can live? I mean, it's even any intellectual life. And any intellectual life forms even very specific, special environment. Therefore, if you make a measurement, you see the very, very special environment, okay? And similar things happen in the universe. In this case, in the different universes, the energy density of space is different. And if you trust this string theory thing, even the species of elementary particle is different. So in different universes, you don't even have electron, you don't have photon, or any of those things. And most of them, at least 10 to the 120, our probability for us to exist is more than 1 in 1 to the 120, 120 zeros. Okay? So it's an incredibly lucky thing that even the galaxy or star forms. That's the current picture of the fundamental physics is telling us. And then we also have there are a couple of ways to even test observation, indirect test. Of course, you can't directly go to a different universe, unfortunately. But yeah, we'll be discussing all of those things.
Right. So that leads me actually to a nice question that someone asked from the audience, and which is, are there any prospects for getting actual observational evidence in some way for the different multiverses that you're proposing? Yeah. Can we actually go and look for them in some form?
Okay, yes. First of all, this is the only theory we have to possibly understand the size of observed dark energy. That's why I started from. This is not some imagination, random imagination to, there may be multiple universe. Then, of course, any comic writer can do. So we discovered in 98 this fact of the vacuum energy density so close to the energy density of matter. There is absolutely no way to understand that fact. And this is the only way we could come up naturally to understand and look back equation, which was discovered in 80s called string theory. This whole mechanism was already contained. So we have to take it seriously, okay? So in some sense, this is the first evidence, okay? We had the vacuum energy, not zero, but it's comparable to the energy density of matter. But then we are talking about this multiverse in very specific contexts, like the bubble universe formation and so on. First of all, for example, something called curvature of our universe. If you just draw a triangle, then this theory tells you that this is a negative curvature, meaning the sum of the three inner angles is more than 180 if you make a big triangle in the universe. Although you don't know how much, so maybe 179.999 or something. But that means that if you, in the future measurement, discover the positive curvature, like 180.1 plus minus 0.03 or something, you can even exclude the theory. Okay, so this is one of the criterion of scientific theory. if the future measurement doesn't get along with this theory or prediction of this theory would be dead. Okay, whatever the Yasunori Nomura told today is just garbage. Yeah, forget about it. So this curvature and then other things is signatures of different universes colliding and so on. But unless we take it seriously, we can't even come up with the signatures. And then the observational people won't look at that, and we would miss tremendous opportunity to learn the fundamental physics.
Thank you, Yasunori. So that's all the time we have for your presentation. It's been truly fascinating. And so with the little bit of time that we have left, what I'd like to do is invite all of our speakers back for a few last questions. Okay, so it looks like everyone's back. And so what I'll start with is a question for Craig, which is, so there's 3 billion base pairs of DNA, and what fraction of that do you think is actually functional or has some relevant information that's useful for biology? I know you maybe answered a little bit of that in the chat, but if you could summarize those thoughts again.
I did type it out in the chat, but I'd say certainly at least 5 to 6% is exonic and evolutionarily conserved and for sure functions. But it's very well known that functional enhancers are not necessarily evolutionarily conserved. So yet 80% of the genome participates in a biochemical event. I think most people think that number's too high. So I think somewhere in between 5 and 80. But I think that's kind of wild that range is so huge. So I think this is really exciting that there's a lot of work to do. My gut feeling, if I had to bet, would be about 10.
Okay.
We don't know.
Right. So it's variable, but a large fraction. Yeah.
I think it's also hard.
So,
it's like absence of evidence is not evidence of absence. So just because if DNA is tested, it doesn't have a function, it's hard to rule out that it has a function in some environmental condition that wasn't tested.
And so now thinking about DNA and moving maybe to multiverses, which is kind of going up many layers of organization here. This is a question for Yasunori from the chat. What do you postulate makes up dark matter, dark energy in our universe?
Okay, if this picture is right, dark energy is nothing other than the energy of the space. That's precisely this is precisely the motivation to introduce multiverse, to understand this dark energy as the energy of space. And dark matter is an elementary particle. That will be the property of our own universe. If you go to the outside our bubble, then what we call dark matter doesn't exist. Even electron wouldn't exist, the photon doesn't exist, because if the shape of the extra dimension is different, then the species changes. So dark matter is our own property, like a property of the Earth. It's not applicable if you go to Mars. And dark energy, being energy of the space or something, or quantum mechanics, is like Newtonian mechanics, which applies even if you go to Mars. If you go to the Jupiter, it applies more fundamental.
Okay, great. And so I think we maybe have time for one question, and I'll pose that to Dan. So we've heard about dark matter, we've heard about junk DNA, and then there was a question in the chat by, I think, a 9-year-old who asked, what's your favorite alien and why?
I'm a big fan of Star Trek. That was a big influence on me. It's probably why I'm doing astronomy and doing SETI. And in Star Trek, when I was Gibson's age at 9 years old, I think I was watching it, and they had a creature, an alien that was made of silicon, not carbon. And I was fascinated by that idea that maybe life didn't have to be based on carbon the way that you and I are based on. And I think that creature was called the Horta. And anyway, Star Trek had a lot of interesting ideas, and I think it influenced a lot of scientists, people that became scientists. And I think it's one of the reasons I got interested in astronomy and this question, are we alone? A more recent movie that I like is Contact. It's about how to find aliens, and it was written originally a book by Carl Sagan. Carl worked with us on SETI. He was an amazing scientist and really good at talking to the public, getting people excited about science. And he wrote that book called Contact, which turned into that movie. I recommend that movie to Gibson and others.
Thank you. Thank you, Dan. Thank you, Yasunori and Craig. And I'm afraid that's all the time we have. Once again, I'd like to have the audience thank all of our speakers for their presentations. And to close, I'm going to hand the floor back over to Dean Kahn.
Thanks, Dipti, for moderating, and thanks to all of our speakers for their compelling presentations. It's always fascinating to hear about the groundbreaking research emerging from our departments. I think many of you are aware that science, in particular fundamental science or basic science, is under serious threat right now. We're facing enormous cutbacks in funding for basic research, both nationally and at the university. Some people ask how they can help. I think the most basic thing is to, make yourselves heard as a voice for the importance of basic science. And also at Berkeley, of course, we will face significant financial difficulties. So those of you who are philanthropically inclined, there are many opportunities to help us there, and we would certainly appreciate it. I'd hope you all will join us on Wednesday, May 21st at 5 p.m. for the next installment of Basic Science Lights the Way, where we will discuss advanced research on the science we all learned in high school. Do you remember sitting in your high school science class wondering, when will I ever use this? Join us as our experts share how we use the basic scientific topics of photosynthesis, gravity, and cell division to uncover fascinating truths about the world we live in. Please reach out if you'd like to learn more about anything we've covered. The video of today's session will be available at basicscience.berkeley.edu approximately 2 weeks from now, and you can always visit the website to watch episodes you missed. Thank you for being part of our efforts to uplift scientific research and education, connect curious people to science and ideas, and support discoveries big and small. Until next time, stay curious, fiat lux, and go Bears.